Hi everyone. So today we're going to learn about the formation of collagen. So if you do not know what exactly is collagen, I request you to go back to my previous video and then learn about the structure of collagen and then come back to this video so that you can understand the basics. Now let us see the synthesis of collagen. So when we talk about the synthesis of collagen, there is a gene which is present in the obviously in the DNA, right? And then the DNA will be converted into RNA, right? And then this process is known as transcription. And then after the RNA is like RNA is made, then RNA will be translated into protein. So as you can see over here, that translation is taking place on ribosome. And so now after translation, the protein which is formed is known as pre-procollagen, right? Now pre-procollagen, after this formation of the simple protein, the hydroxylation of proline and lysine will take place. That means an hydroxyl group will be added in the proline and the lysine. So this is in the fourth position and this is in the fifth position. Now after the hydroxyl group is added to proline and lysine, what will happen is it will be either it would be either hydroxy proline or hydroxy lysine, right? So these are like the two things. So the reason why a hydroxyl group is being added to the pre-procollagen is to increase the stability. That means if an OH is present, then what will happen is there is a possibility of making a hydrogen bond. There is a possibility that one simple helix will be able to form hydrogen bonds, like intermolecular hydrogen bonds with one another. So as I told you, if you don't understand it, please go back to my video and then learn about the hydroxyproline and hydroxylysine. How does it look like? So I'm just gonna go like simple one on this one. So after the formation, like after the hydroxylation and formation of hydroxyproline and hydroxylysine, this particular protein pre-procollagen will come out in the cytosol. And then when it comes in the cytosol, the process called glycosylation takes place. So what exactly is glycosylation? Is actually addition of sugar moieties. So the sugar moieties would be galactose or it would be glucose. And then if I talk specifically, like if I have to tell you on which amino acid it gets attached or it gets connected, that means it's glutamate. So glutamate is actually an amino acid which has a carboxylic acid in its R group, right? So this is the one. So glutamate. So after the formation of after the glycosylation, now this pre-procollagen will be known as procollagen. So as you can see, the name is also changing, right? And then afterwards, these procollagen will come together. That means 3 procollagen will come together and they will form a triple helical structure. And then after that, what will happen is there is an enzyme called procollagen peptidases. So this procollagen peptidases, what it will do is it will chop off the ends of the N and the C terminal, right? So why is it chopping off? Because as you can see over here that the helix structure is not properly formed. In this particular direction also, as you can see, the helical structure is not properly intertwined. And why do we have to keep this underwined or not totally like triple helix formed structure, when we can like cut it, right? We can cut it and we can make a proper structure. So this enzyme comes and cuts at this direction, and then afterwards what will happen is this particular structure will be secreted out of the cell. it will reach the extracellular space, and then that means it is coming out of the cell now. I hope you're able to understand. It is being secreted, right? After the removal of N and C terminal domain, now what will happen is, this will be known as trophocollagen. That means this procollagen peptidase is actually working when it is being secreted outside the cell. So don't get me confused. Like, I'm just trying to say that this will be like remain like this only. Just come out, and then only the removal of N and C terminal will be cut off, right? Now this particular structure is known as tropocollagen. This structure is actually the structural unit of collagen. Let me just write. So this is the structural unit, right? Although it is not yet functional, it is just the structural unit, it is not yet functional. So this is the structural unit, tropocollagen. Now what will happen is, in the next process, cross-link formation will be there. That means this is a tropocollagen, this is a tropocollagen, this is a tropocollagen, tropocollagen, tropocollagen. Now all these tropocollagen with cross-link with one another. Now you must be wondering which is the which is the amino acid which helps in the cross-link. So lysine is the amino acid. So lysine, they'll form cross-link with one another, and then they'll form a long structure of collagen fibril, right? This collagen fibril, as you can see. So this will connect with one another, and they'll form a long structure called collagen fibril. And then this will actually be the structural unit, functional unit of collagen. That means now it is functional, now it can perform some functions, okay? So this is actually the synthesis of collagen: translation, hydroxylation, then glycosylation, then triple helical formation, secretion, then removal of N and C terminal, then cross-link formation, and then what are the amino acids which are actually being involved in those processes. You have to know about it. So hydroxylation is usually in the proline and lysine and the 4th and 5th position, right? So this is all about the collagen synthesis. If you like this video, please like, share and subscribe. Thank you so much for watching. Hi everyone.